Okay, good evening. It's still afternoon. Sorry for the delay in starting the
Good evening,
tutorial.
sir.
I've had a few network problems. Are you able to get me?
Sure, sir.
Sorry, are you able to get me?
Yes, sir.
Are you able to able to get me?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, people are awfully quiet. I need the response so that we can proceed.
We are able to get you, sir.
Okay, thank you very
We
much.
are able to get you, sir.
So, yes, our tutorial, we'll start with the biology one. Okay, let me just share the notes. So we're looking at nucleic acids. This is something that we already know, but we can just highlight a few points. So feel free again to, whenever there's something that you don't understand, you quickly come in or you quickly ask so that we proceed together. Right. Are we able to see the screen?
Yes, sir.
Yes, sir, we can.
Okay.
We are able.
All right. So nucleic acids are basically chemicals that detect the amino sequence in proteins. Most of the structures of our body are proteins, and for those proteins to look the way they look, we need amino acids. So this is what we'll go phase by phase and try to fuse in some questions as we go on, so that we cut on time. We already lost a few minutes from our time. So they give information to chromosomes, which is then passed from parents to offspring. Okay. So the central dogma, which you know, so there is DNA, okay, RNA, then so from DNA to RNA we have transcription, from RNA to protein we have translation. From DNA to RNA we have transcription, RNA to protein we have translation. Okay, so there are two types. We have RNA, which is ribonucleic acid, and then we have DNA, which is deoxyribonucleic acid. If you've seen, the difference starts first of all in the name. Deoxy means there is no oxygen. Okay, so deoxy is lacking an oxygen. RNA has the oxygen present. Okay, so we'll look at this in detail, but from here we can get the difference there. So deoxy is minus the oxygen. Okay, so the basic principle or nucleic acids are in two forms. We have nucleotides and nucleosides. A nucleotide has nitrogenous base, pentose, and phosphate. The nucleoside only has a nitrogenous base and a pentose. When there's no phosphate, it's a nucleoside. When there's a phosphate, it's a nucleotide. So you can get that difference first. As you see your MCQ questions, or as you've already seen the MCQ questions, you should be able to differentiate a nucleotide from a nucleoside. Okay. What are they made of? Nucleotides. Okay. We talked of a pentose sugar. The pentose sugar, this pentose sugar is a 5-carbon sugar. So there are 5 carbons. Then we have nitrogenous base, and then we have a phosphate group. Okay. So the phosphate group is what differentiates it from the nucleoside. So examples of nucleic acids, we've seen them there. Okay. The nucleosides and the nucleotides. Okay. Okay. So again, nucleic acids, a diagram on the bottom right is showing us the difference between or the structure of the nucleic acid. So we have a purine base or pyrimidine.
And the sugar.
Okay. So please mute your microphone. Okay. So we have the purine base, then we have the pentose sugar and the phosphate. So this is just emphasizing on the structure. So again, structure, 3 components. Always remember these 3 components: a purine or pyrimidine base, a pentose, and then a phosphate. Okay. So structure again, ribonucleic acids. Remember we said they have a hydroxyl group at position 2 and position 3 is the hydroxyl group. Then the deoxyribonucleic acids have the hydrogen at position 2. So we'll look at the diagram where we'll see, okay, so we have a nucleotide here, we have a phosphate, and we have a purine or pyrimidine, which is a nitrogenous base. Okay, so this is a monophosphate because there's 1 phosphate group. When we add another phosphate group, we have 1 phosphate adenosine monophosphate. An example is AMP. Then if we have another one, it becomes a diphosphate. If we have another one, it becomes a triphosphate. So adenosine triphosphate or ATP has 3 phosphate groups bound to the nitrogen glucose molecule and the nitrogenous base. So you need to look at, or you need to know at which positions is the bonding. So at carbon 1, that's where we have the nitrogenous base. And at carbon 4, we have the phosphate group. Carbon 1, carbon 4. Okay. Okay, so it determines when we have 1 phosphate, 1 sugar, and 1 base, we have a nucleotide, like what we are seeing here. Okay. So the sugars that I use, we have, depending on the number of carbons. Number of carbons gives you the sugar used, and it also determines the position at which the bond will take place. Okay. So a ribose. Sorry, someone saying something. Okay, so let's look at deoxyribose. At position 2, it's only a hydrogen. In the ribose, we have the hydroxide at position 2. That's the major important difference. So up there again, so concerning the sugars, just the position and the number of carbons that you need to know. This is a 5-carbon sugar, so it's a pentose. Okay. Then the types of nitrogenous bases, we have purines and pyrimidines. Purines, example of purines are adenine and guanine. These have 2 rings each. Adenine has 2 rings, guanine has 2 rings. Then the pyrimidines, we have thymine, cytosine, and uracil. Okay. Okay, so sorry, apologies, we'll slow down just a little. We are trying to work within the short time, but I think we can still cover. Okay, so please, is there any point which is not clear, we go back, or how far should we go back? How far should we go back? Anyone who's not clear?
You can proceed.
To proceed. Any other people?
Can continue, sir.
Okay.
Can continue, sir.
So we'll slow down just a little so that we have everyone here. Okay. So again, the major importance we are seeing in this diagram is one, the sugar has we are seeing 5 carbons on the sugar. Okay. On the 5th carbon, that's where we have the phosphate binding. Okay. On the 2nd carbon, that's where we determine whether it's it's DNA or RNA on the 2nd carbon. Okay. So the 5th carbon, we have the phosphate. 2nd carbon, we determine whether it's DNA or RNA. 1st carbon, we have the nitrogenous base. Okay. So nitrogenous bases, like I said earlier, are in 2 forms. There are those with 2 rings and there are those with 1 ring. So those with 2 rings are called purines. Those with 1 ring are pyrimidines. Examples of purines, we have adenine and guanine. They have 2 rings each. Pyrimidines have 1 ring each. So remember, like we did under the chemistry of water, we looked at the hydrogen bonding and what causes hydrogen bonding. So even in this case, we have hydrogen bonds. How these hydrogen bonds operate and how they interact, will relate to them later on as we proceed. Otherwise, we're supposed to, or for now, we're supposed to remember that the hydrogen bonds are also applicable here. We shouldn't forget about that chemistry then. So nitrogenous bases in nucleotides consist of 2 genotypes, which we've already talked of. Okay. So purines, adenine is abbreviated as A, and guanine is abbreviated as G. Then pyrimidines, cytosine, C, thymine, T, uracil, U. So these are points to remember. Okay. So thymine is only found in DNA. This is a key point. This is a pearl, something you shouldn't forget. Thymine only found in DNA. In RNA, thymine is replaced by uracil. So whenever you see a sequence and you see the letter U, remember that is RNA. Wherever you see the letter U in the nitrogenous bases. Wherever you see the letter T, that is DNA. Otherwise, the two are structurally similar. Okay. So RNA, DNA. The top part, we see RNA. We are seeing a U. Remember, U is only for RNA. T is for DNA. The rest are the same. Okay. So now DNA. DNA, when untwisted, occurs in pairs. It has 2 strands. You have the lagging strand, okay, and the leading strand. The structure or how these strands come about, we'll look at them later in another topic when we look at replication and translation. For now, we're supposed to know the structure of DNA. It has 2 strands. Okay, we have the leading and the lagging strand. So on our far left, we have this strand here. Okay, what we have is that the sides of the ladder, imagine this as a ladder. Okay, the sides of the ladder have a phosphate and a sugar molecule. Phosphate, carbon number 5 of the sugar molecule. Okay, these are the sides of the ladder. Then the steps in between the ladders, okay, where you step, that's where we have the nitrogenous bases. If it's DNA, we are talking of C, G, T, A. These 4 bases are found in DNA. Okay. This diagram is showing us how they pair. C binds to G. A binds to T. Okay. Just like that. So C and G, A and T. There's no 2 ways about it. It's just that way. Okay. So we have adenine, thymine. Okay. A always pairs with T. Okay, apples are tasty for those like mnemonics, cytosine, and guanine. CO is pairs with G. Cookies are good. Any questions thus far, or any questions so far? Our session will be cutting in a few seconds, so feel free to rejoin.